Good evening, Ishika. Okay, how are you doing? You got the flu shot, right? Last time, influenza. Today only a news broke out that you can say the SARS-CoV-2 has got 17 mutations in one strain at one go in United Kingdom. And one of those mutations is pretty, you can say, lethal also. It increases the transmissibility of SARS-CoV-2. N501Y at 51st position, asparagine has been changed to tyrosine, you can say, missense mutation. At 69, 72 nucleotides have been deleted in the spike protein sequence, which makes it more, you can say, gives it more affinity towards binding with the H2 receptor. Maybe the vaccines may not work against this strain. That's the thing they're trying to find out. Now, UK is under again lockdown. Indians have cancelled, like, there has been ban on UK flights in India till the end of this year, 31st December. And the work on universal influenza virus vaccine is also underway. It has shown quite promising result in phase 1 study in mice model, so that we don't need to get it again and again. Because influenza virus, it mutates a lot. Its mutation rate is very, very high, so you need to get it each year as per the current strain. That's the work that they're doing. So last time we had talked about the various transcription factors, like the TF2D, TF2H and all, TF2A, then downstream promoter sequences, then the specific motif 10 element, then derecognition element BRI, then the INR box, INR sequence, then the Tata box, right, at minus 25. So two things, this TF2H is great thing, like it has got the dual helicase activity. I told you XPB and XPD subunits it has. So what it does is, you can say, the TF2H can catalyze the breakage, also means the denaturation. The helicase activity is bestowed upon the RNA polymerase 2 in eukaryotes by TF2H. You can say transcription factor, and then and the main of that, the most important of that is XPB, because XPB subunit will catalyze the 5 to 3 prime helicase activity, then kinase activity, then DNA repair activity, right? So if you write in a summary or in a specific, you can say, point-wise manner, flowchart manner, you can say that the phosphorylation on serine 5 phosphorylation means phosphate group attached on serine 5 of RNA polymerase 2 in case of, you can say, or by catalyzed by TF2H. This causes the recruitment of the enzymes that cause the capping. Okay, I'll explain to you what is capping in just a few moments. That is 5-cap enzyme recruitment. And then we also talked about the elongation factor is the PTEFB in case of the prokaryotes. So that causes phosphorylation. Transcription elongation factor B, that causes the phosphorylation on serine 2. Okay. Serine set or present at the amino acid number 2 in again RNA polymerase, that is caused by PTEFB, and this results in recruitment of, you can say, elongation, splicing and polyadenylation factors. Other elongation, then we have talked about splicing, and we'll also describe polyadenylation. Factors means proteins. So these are the two main events that occur. Okay, there are various other events also that take place. So phosphorylation on serine 7 that happens, then dephosphorylation of serine 5 that occurs, right? So you can say those things they keep on going on and they help in the, you can say, recruitment of poly A factors, poly A polymerase. Okay, so those things keep on happening, but this is enough as far as your course is concerned. And then what happens is we have something known as, which is very important, capping and tailing. Okay, capping and tailing, because capping and tailing are extremely required. Why? Because if they do not occur, okay, if they do not occur, the mRNA which has been produced in the nucleus, okay, may get completely degraded, completely broken down into fragments in the cytosol before it even reaches the RER or the ribosome present on the rough endoplasmic reticulum or endoplasmic reticulum, making it RER. Okay, so it may never reach because there are 3' exoribonucleases, there are 5' exoribonucleases, and there are all also present known as endoribonucleases, which can trim the mRNA in the cytosol as soon as it is exported from the nucleus, a double membraned nucleus. As soon as it gets, you can say, exported, it may be completely broken down. Okay, and when it is broken down, it is of no use. Okay, it is of completely no use. It will become a useless entity, and therefore no protein can be produced. Okay, no protein can be produced whatsoever. Okay, so the thing is, the 5' end of eukaryotic mRNA, first of all, that needs to be protected. Okay, so that there is complete protection from the 5' exoribonucleases, because in the cytosol, okay, in the cytosol, suppose this is the nucleus double membrane, you have RAN GTP mediated export, that is not in your course. Suppose mRNA has been exported, hnRNA has been converted to mRNA. Now mRNA is exported, but before the export in the nucleus itself, mRNA needs to be protected at the 5' end and at the 3' end both. Okay, so that is the essence of protection, so that it is protected from exoribonucleases of two types, 5' and 3'. Okay, still it is vulnerable to endoribonucleases. Sometimes it can be cleaved by endoribonucleases, but it lowers the probability of its cleavage, okay, or of its degradation by many folds. These kinds of protection from both the exoribonucleases, 5' and 3' exoribonucleases, are specific for those polarities. Now, how does the capping takes place? Capping means you will attach a molecule at the 5' end of RNA. Like you wear cap where you don't wear a cap, or you don't put on a cap on your leg or on your toes, right? You put it on your head. The head is the starting, the initiator point of the body. So the initiating point is the 5' end. So whatever protection happens here is known as 5' cap, and tail means like you have your toes, that's the end part. So whatever protection is here, that will be known as 3' tail or simple tail, is also called as capping and tailing. Okay, capping, tailing, and simultaneously you will go for splicing as well. All three processes, all of these three processes happening in the eukaryotic system, they occur simultaneously. They do not wait for the mRNA to completely end its transcription. As soon as the first few nucleotides are produced, the 5' is capped and splicing continues, and as soon as the mRNA is terminated, you will see the polyadenylation continuing. The factors will already be recruited, right? and they will do some differential polyadenylation also, because there will be various polyasites. Some may be chosen for one RNA, some may be chosen for another RNA. Okay, so that is again a point of difference. Okay, so we won't delve into that. So the thing is capping, tailing, and splicing. Okay, these are the three post-transcriptional modifications. Okay, known as PTM, post-transcriptional modification. So now we'll be studying in detail about capping. So you can say that transcription starts with a nucleotide triphosphate, like usually adenine or guanine. Now the first nucleotide retains its 5 triphosphate group, okay, and makes the usual phosphodiester bond from its 3 position to the 5 position of the next nucleotide. So that concept you must be having, because the first it can go for de novo polymerization that I told you. It doesn't need any primer, the RNA polymerase. So first one, suppose it came as ATP. So there will be ATP, first will be your A, it will come in the form of ATP and you will not need any hydrolysis. Only when the second one comes, suppose GTP, it will hydrolyze into GMP plus PPI plus energy, and this energy by the dissociation produced by the dissociation of the gamma phosphate bond will be attached to this one. Okay, so here you have 3 phosphates, here you have only 1 phosphate, like that. Okay, so first nucleotide will always retain all its phosphates, so it is a triphosphate. That's the first concept that should be there in your head. Next, you can say that the addition of, okay, 5 prime terminal guanine is done by a specific enzyme, okay, known as guanylyl transferase. Okay, so in experiment it has been found that the 5 prime end, okay, does not give rise to, you can say, expected nucleoside triphosphate when, you can say, the mature mRNA is treated in vitro with enzymes that should degrade it into individual nucleotides. Okay, so then scientists found that why is it happening? Why not the RNases are able to break down, exoribonucleases are able to break down it? So, or why are they not able to break it down? So what they found was, instead the 5 prime contains 2 nucleotides that are connected not by the 5 to 3, okay, triphosphate linkage, but it is connected by a 5 to 5 prime triphosphate linkage and also bears a methyl group. Means here the guanine one, okay, the guanine, you can say, is added with, or the GTP is added to this one via a 5 to 5 prime linkage, not 5 to 3, and also a methyl group will be subsequently added to this one. So you cannot break it down by 5 prime exoribonucleases. Okay, you cannot break, you cannot start the cleavage here. Okay, so that is the event that happens. Okay, now this structure is known as, you can say, the 7-methyl guanosine cap. Okay, so the reaction that happens is, this is the reaction that happens. 5 prime, you have already, and write it down for you. This is guanine, and you have 3 phosphates like GTP. I'm only writing the phosphates here. Okay, then you have here 5 prime, A has 3 phosphate. Okay, then you can have anyone, like you can put it as N, that will be one phosphate, suppose G, this is C, okay, again N. So rest are all monophosphates, okay. Now what you will get is these two phosphates will be now taken out, okay. These two phosphates here will now be taken out. You will get 5 prime G, okay, triple phosphate attached to 5 prime to 5 prime linkage. Yeah, I can write it here, maybe it will be better. 5 prime to 5 prime linkage, okay, to your adenine. Here only one phosphate, these two phosphates are gone, okay. Then the rest will be the same. Other nucleotides, two phosphates are gone. So here you can say, so this is the 5 to 5 prime linkage, okay, and later on you will be having a specific methyl group here as well, okay. So this is what the reaction is, and the reaction and this structure is known as the cap, the 5 prime cap, okay. This reaction is known as the 5 prime capping, okay. So always be sure of this specific sequence, okay. So the thing is, if you see it stepwise, okay, I'll write it down stepwise as well for you. So the first reaction is catalyzed by an enzyme known as, you can say, guanylyl transferase, okay, guanylyl transferase. So first of all, what you will see is you have the triple phosphates, okay, you have triple phosphate here, okay. So A, you can say triple P, then N, P, then NP. So first enzyme will be a phosphohydrolase, okay. What it will do is it will take out the gamma pi. This is alpha phosphate, beta phosphate, gamma phosphate, okay, connected to the first nucleotide of the mRNA, okay. So what will remain is, now you have, suppose this one, two phosphate groups are remaining, okay. So you will be left with alpha phosphate, beta phosphate, and now comes your guanine with its alpha, beta, gamma, okay. Now what will happen is you will be able to achieve the attachment of 5 to 5 linkage via guanylyl transferase. Now in biochemistry, you have a nomenclature rule, okay, guanylyl or adenyl transferase and adenine transferase. What is the difference? Difference is, I told you once when I was teaching you DNA, that adenine, adenosine, adenylate, there is a specific difference. Difference is, this is only a base, this is base plus ribose sugar. In case of DNA, deoxyribose sugar, RNA ribose sugar, this is base plus sugar plus phosphate group. Means this is a nucleoside, this is a nucleotide. Now, guanine is same. So guanine, guanosine, then guanylate. So guanylyl transferase means you will be transferring this one, GMP. If it is guanine transferase, you are transferring only base. Adenine transferase, you are transferring only the base. Meaning that this guanylyl transferase will do double job. It will take out these two phosphates and also attach this one. Okay. So what will happen is, if you erase this, what will happen is you will have guanine. Now it is triple phosphate. These two phosphates are gone. One phosphate of guanine, two phosphates attached to the adenine. They remain, and you have this one and this one going on. Okay, and the linkage is, you can say here, 5 to 5 prime linkage that I had showed you. This is done by guanylyl transferase. Concomitantly, two phosphates is released, one phosphate here, two phosphate here. Total 3 phosphates have been released. Okay, now you need a methyl group. So at the 7th position of the guanine, okay, guanylyl, spelling will be this one, guanylyl 7 methyl transferase. Now it will transfer the methyl group to the, you can say, 7th position, okay, of guanine. So it will now be called as M7G triple P. You can say now A, like that. So now you have A, then you have NP, NP, and so on. So that will become this one. Okay. So this is your 7-methylguanosine. Sometimes what happens is, at the second, or you can say this is on the base. Now what happens is, on the second carbon of the ribose sugar of guanine, this is done on the base guanine, but sometimes second carbon, since I put a prime here, second carbons, okay, on that oxygen that is attached to the second carbon, that methyl group is again transferred. Methyl transferase. Okay, so then that is also done, and then you have more, you can say, protection. Okay, so whenever you add prime, okay, if you have I'll show you the diagram of a nucleotide, it will become clear that why do we add prime. Okay, so if you see nucleotide structure, okay, just look at this nucleotide structure. Okay, so you have carbons here, right? You have carbons here. Wherever there is no nitrogen, here is carbon, here is another carbon, here is carbon, here is carbon, and this is also carbon, carbon 1, 2, 3, 4, 5. Okay, how will you distinguish between the carbon present on the ribose sugar and on the base? So historically, nomenclature wise, this is a very very important rule that most of the, none of the books, I think, they have put it clearly. The thing is, conceptually what is there is, the carbon, if you are referring to the carbon present on your belonging to the ribose sugar, this deoxy or simple RNA, you can say ribose sugar, whether you have oxygen here or not. So since this is lacking oxygen here, so this is a deoxyribose sugar, this is a DNA one. So at the second carbon, you don't have oxygen in the DNA, right? So if you are looking at a molecule like this, it means that if I am writing 2 prime, okay, or 3 prime, 5 prime to 2 prime, what do I mean? I mean that that carbon belongs to the ribose sugar. 5 prime means fifth carbon. 1, 2, 3, 4. 1, 2, 3, 4, 5 means I mean this carbon. 3 prime means I mean this carbon. And if I'm not writing prime, 7-methyl guanosine means seventh carbon. You have to count here seventh carbon. Okay, that means the carbon belongs to the, you can say, the base, because phosphate group does not have any carbon. So in order to distinguish between the carbon present in the base and in the ribose or deoxyribose sugar, we are using the prime, you can say, nomenclature. When I am saying prime, I mean that the carbon belongs to the base, ribose base. Okay, sorry. When I am using prime, I mean that the carbon belongs to the ribose sugar, whether it's deoxyribose sugar or simple ribose sugar, that is this pentose sugar. Okay, it belongs to this pentose sugar. When I'm not using any prime, I'm just adding 7 carbon, I mean that the carbon belongs to the base, adenine, guanine, cytosine, thymine, or uracil in case of RNA. Okay, so this is the way the capping event occurs. Okay, so is there any doubt in the capping event and in this nomenclature, Ishika? Prime one, because none of the books they provide the information regarding the prime one. Okay, that is the greatest problem that they face. Okay, good. So now we come to the, you can say, the tailing event. Okay, tailing event. So we have done the capping. Splicing is left. Okay, splicing is not given in your course in detail. So we'll skip the splicing machinery. If I have time later on, I'll explain to you splicing as well. It is very interesting how does the splicing take place. Okay. So now we'll switch our focus to tailing. Okay. So in splicing also we have spliceosomal splicing, non-spliceosomal. Then snRNPs, that is small nucleolar ribonucleoproteins U1, U2, U4, U5, U6 and all. So we'll not go into that. Group 1 introns, group 2 introns. Okay. So now we'll switch our attention to, you can say, splicing. Okay. So sorry, the tailing. So alternative splicing also I had explained to you in a brief way that how do we produce 1 lakh proteins from only 22,000 genes, right? So now we come to tailing. Now what does tailing mean? Okay, tailing means we add 2 to 300 adenine residues, okay, to the 3' end. 2 to 300 adenine residues to the 3' end. Now what has happened here is, when you have done all this to the 5' group, this is 5 to 5' linkage. Now when the 5' specific RNase or ribonuclease comes, it just cannot detect because of the bulky methyl group. Methyl group causes steric hindrance. Okay. And 5' RNase will try to do what? Will go for the 5 to 3' bond searching. That is why the addition is 5 to 5' linkage here. So that it will go for detection of this phosphodiester bond, and then it will cause the cleavage. But even if the steric hindrance cannot completely eradicate its binding, it may try to bind. You can say not very tightly, but binding can happen sometimes. Still, the 5 to 5' linkage will not fit into its active site, or its active site cannot catalyze the cleavage of the 5 to 5' phosphodiester linkage. Okay, the orientation is different. So it will directly fox or outwit the 5' RNases, exoribonucleases. Okay, 5' and 3' both. Neither it is a 3' nor it is a 5'. So it is providing a specific protection from both types of exoribonucleases when it comes to capping. Okay. Now when it comes to tailing, what you do is you have a 5 to 3' end. and we add 2 to 300 adenine residues. So we have a specific poly A site present in the DNA, present in the antisense strand, and the complementary is made on the RNA that I explained to you via on when I was explaining to you the intrinsic termination that AAA, it will be trimmed off. Okay. So what we do is then we go for, you can say, the specific poly A tailing later on. Okay. So now this AA will do what? This is 5 to 3 prime linkage, so this will already provide protection against the 5 prime RNA or 5 prime exoribonuclease because 5 to 3 prime is there. It will come from this side, not specific. 3 prime exoribonucleases will have to cleave this A. So many A's are there, it will provide it ample amount of time to go and bind to the ribosome before the 3 prime exoribonucleases can come and can while the degradation can reach the real structural gene 3 prime site, because in order to cleave 300 phosphodiester bonds it will really take time, right? So it will provide it ample amount of time to get out of the harm's way, you can say, right? So it is still not clear whether RNA polymerase 2 actually engages in a termination event at a specific site because this is eukaryotes. Okay, you don't have the prokaryotes here. Okay, the intrinsic termination really does not happen. Okay, just I gave you an analogy, but this is eukaryote. So here the dephosphorylation and phosphorylation of the carboxy terminal domain of the RNA polymerase 2 will cause the termination. Okay, here rho factor and all they do not exist. Okay, so keep that in your mind. Now what can happen is the mature 3' end of polymerase 2 transcript mRNAs are generated by cleavage followed by polyadenylation. So addition of poly A to nuclear RNA, you can say, can also be prevented if you add analog of deoxy or 3' deoxyadenosine in your experiment. Okay, like cordyceps you can add, you can prevent it also. Okay, so if you prevent it, it will be very susceptible or vulnerable to the 3' poly A tail, or sorry, 3' RNases present in the cytoplasm or cytosol, and will be able to degrade it. Okay, will be able to easily degrade it, right? Now you can say, what you can say is the site of cleavage or polyadenylation in most pre-mRNAs, means which have not been, you can say, added or which have not been modified by polyadenylation, because polyadenylation will take time to occur, so you can call it pre-mRNA. Okay, it is, you can say, this is a sequence known as AAU-AAA. Okay, in case of RNA, if you have a sequence of AAU-AAA, this is the poly A site. Now this sequence will act as a recognizing entity, okay, for the polyadenylation machinery to come in, and then it will cause the catalysis of the poly A tail formation. Okay, so the AAU-AAA site is needed for cleavage and pollination because one more thing is, in experiments, deletion or mutation of the AAU-AAA 6 nucleotide, this is, so you can call it an hexamer. It prevents generation of the polyadenylated 3' end. Okay, that has been seen in research. So it is the, you can say, fundamental indispensable sequence needed for the polyadenylation to occur. Now what happens is, generation of proper 3' terminal structure depends on The proteins like number one is CPSF, cleavage and polyadenylation specificity or specific factor. Okay, first this protein will come here and bind CPSF. Okay, that is cleavage and polyadenylation specific factor. First this protein will come in and bind, and it will recognize the AAU AAA, okay, and also, okay, will bind to the another factor, okay, known as cleavage stimulatory factor. So this will bind to AAU AAA and will also bind and stimulate, you can say, CSTF, cleavage stimulatory factor. ST means stimulatory, F means factor. So you have CPSF and you have CSTF. Okay. Now these components will bind directly, okay, to another sequence which is a GU rich sequence. Okay. So in an mRNA or pre-mRNA, there will be a sequence like this. Till here, structural gene is over. Now you have AAU, AAA. Now after this also there will be a GU rich sequence. Okay, when CPSF and CSTF have bound, CPSF and CSTF have bound to AAU, AAA region. Okay, now one of these components will bind directly to a downstream GU rich sequence. Okay, and then it will enhance each other in recognizing various polyadenylation signals. Okay, they will bind here and then they will further activate themselves. Okay, they will activate themselves and act like endonuclease. Okay, and cleave the RNA, and they will cleave the RNA. Okay, they will completely cleave the RNA at somewhere between, you can say, GU rich and you can say AAU-AA sequence. They will now, this CSTF cleavage will be activated after binding to this half activation, after binding to this full activation, and then somewhere here the cleavage will happen. Okay, somewhere here cleavage will happen, and then recruitment of poly A polymerase PAP will come in. Poly A polymerase will come in, and next step will be 5 prime to 3 prime end structural. This is the AAU, AAA, this is CPSF, this is CSTF. Okay, now you have PAP. Now from here you will be adding A. 2 to 300 AAs will be added. Okay, they will, the polymerase is a specific RNA polymerase which has non-specific catalytic activity, means it will only recognize, okay, the ATP as the substrate, because if it recognizes GTP, CTP, it will add all types, but it will only recognize ATP as the substrate. And in the beginning, first 10 residues are added to the 3' end. Okay, it is really dependent on the AAU AAA sequence. Okay, in the second phase, you can say there is another protein known as poly A binding protein. Okay, and then poly A binding protein PABP, it will come and bind to the, okay, poly A tail to allow the poly A polymerase to keep on synthesizing the tail till 200 residues. Okay, so then first 10 residues will be synthesized. What is it for 6, 7, 8, 9, 10? And then poly A binding protein type 2 will come in, and then more will be bound. In the end, you will be having 200 residues. Okay. So then phase 1 and phase 2 of polymerization happens between catalyzed by poly A polymerase, and then we get complete 200 residue. Okay, polymerize, or you can say poly A tail. Okay, so the poly A polymerase by itself adds A residues individually to the 3' position. Okay, but the problem is, if the CSTF and CPSF they are not present, what would happen is the intrinsic mode of action of poly A polymerase is very distributed, means it dissociates after each nucleotide has been added. Okay, so it will take a lot of time. But the presence of CPSF and PABP2, okay, it increases the processivity of the poly A polymerase to extend the individual poly A chain, and it should not take a huge amount of time. Otherwise, it will take days to go for polyadenylation, and we have to then wait for the protein synthesis for days. Then a cell will die because the protein is over and mRNA is not ready to come out of the nucleus. Okay, that is why after 10 nucleotides, this PABP2 comes and binds, so that now the poly A polymerase does not fall off from the template strand. It can catalyze the activity or catalyze the synthesis of addition of A residues. Otherwise, after each A residue addition, it will fall off again, it will bind again, fall off again, it will bind again, fall off. So it is very non-processive. It is kind of distributive, and when poly A polymerase comes in contact with CPSF and also poly A binding protein 2, then its processivity increases. Processivity means the ability to tether or to hold on to the template strand, okay, while synthesizing the nucleotides or while causing the addition of nucleotides. Okay, processivity enhances. So this is how your tailing is achieved. Okay, so now we have completed the transcription in prokaryotes and eukaryotes also. Essentially, in the next class we will begin the translation process. Okay, so now tell me, Ishika, if you had any kind of doubts or queries in today's lecture. Any doubts, any queries, any clarification needed? So such complexity stays in eukaryotes in order to protect its mRNA. Okay, so the many therapeutic uses are also there. We can disturb it, we can cause mRNA degradation. Then later on we will be studying about microRNA, small interfering RNA that can bind to mRNA in the cytosol and then also can degrade it. It may not let it bind to the ribosome. Okay, so those are also many kinds of therapeutic options are there in cancer immunotherapy and all. Okay then, have a nice day and thanks a lot, and let's meet in the next class, Ishika.